Hello friends. In this video, we will be discussing the questions from the GAT-B paper from 31st question. In the previous video, we have discussed the questions from 1 to 30 of section B. So let's continue. The 31st question is, an infectious RNA particle is known as. So I would like to tell, and here it has specified the word RNA here. So a viroid is an infectious RNA particle which infects only plants. Okay, so this infects only plants, whereas virion is a complete. It can be it is having a protein capsid. It is having the genetic material and the tail fibers and everything. And this genetic material can be DNA or RNA. In the case of virion, and prion are the infectious protein. Misfolded proteins are called as prions, which cause different diseases. And next is virusold. There is nothing like virusold, so all these options are wrong. An infectious RNA particle is known as viroid. Next question. Phosphofructokinase is allosterically regulated by several effectors. Which one of the following would allosterically inhibit the enzyme? Okay, so phosphofructokinase, what we call, is helpful in the conversion of for fructose 6 phosphate to fructose 1,6 bisphosphate. Okay, so here why this all process is taking place? To produce energy in the form of ATP. Okay, so when there is excessive amount of energy already present, so here when ATP is already present, there is no need to activate the process of converting fructose 6 phosphate to fructose 1,6 bisphosphate. Okay, so here whenever ATP is already present, so phosphofructokinase is inhibited when ATP is present. So ATP is the correct option here. AMP and ADP are not the energy sources. So whenever AMP and ADP are present, these are the positive regulators. But here it is allosterically regulating. That means if more ATP, it inhibits. When there is less ATP, it down regulates. Oh, I'm sorry. When there is low ATP, it activates. I'm so sorry. It activates phosphofructokinase. Next question. Several RNA viruses form replication complexes by utilizing membrane from. So the RNA viruses, they have to form the membrane around them. Okay, to infect the other cells, they have to assemble again the genetic material, the protein and the tail fibers, everything. So they use a membrane from the endoplasmic reticulum and they form their own complexes. Next question. Procheck is a. So Procheck is used a tool to calculate the molecular weight? No, it is nothing to do with the calculation of molecular weight. It is actually used for the three-dimensional study of the protein structures. So whenever we want to study the structure, how the protein is having the structure, its three-dimensional structure and everything, we use the Procheck. So it is a tool to assess the stereochemical quality of protein structures. So here 3 is the correct option. Stereochemical quality means to study its actual structure in the 3 dimension. Protein nucleotide databases? No, we have nothing to do with the database here. Okay, which enzyme is involved in the repair of DNA induced by UV light? So what happens when there is a UV damage onto the DNA? It forms, here we see, the thymine have formed the dimer. Okay, so it is very important that whenever there is a formation of thymine dimer, we break that dimer so that the damage is repaired. So an enzyme called as DNA phosphorylase binds onto the thymine dimer and cleaves the thymine dimer. Then it is normally produced again. Okay, yes. So here the answer is photolyase. Photolyase is an enzyme which binds onto the thymine dimer. Fatty acids cannot be converted into carbohydrates in the body as the following reaction is not possible. Okay. Formation of acetyl-CoA from fatty acids. Okay. Yes, acetyl-CoA can be formed from fatty acids. This is correct. So this is a correct because like this happens. So here they are asking not possible. So this is not the correct option. Fructose 1,6 bisphosphate to fructose 6 phosphate. Here you can see in the reaction producing fructose 6 phosphate from fructose 1,6 bisphosphate. This is a reversible reaction. This can also happen. Whereas conversion of acetyl CoA to pyruvate is not possible because this is an irreversible reaction. So this step is not possible. That they are asking not possible. So this is the correct option here. Okay. An E. coli cell culture of 1 ml was diluted 10 to the power 6 fold and 200 microliter of this was used after okay, after 24 hours of incubation, the colony forming units was found to be 150. The colony forming unit count of the original culture is. Okay. For this, first let us write down the formula for calculating the colony forming unit. Okay. Yes. So CFU is equal to number of colonies multiplied by total dilution factor divided by volume of culture plated in ml. Okay, so this is a formula to calculate. So how many number of colonies were formed? The number of colonies which were formed were 150. Then CFU colonies formed were 150. So let's write down here 150 into total dilution factor. The total dilution factor is 10 to the power 6 fold. Means 1, 2, 3, 4, 5, 6 divided by, here they have given next is volume of culture plated in ml. So volume of culture plated is 200 microliter it is given. So 200 microliter means 1 liter is equal to 0.01, sorry, 1 microliter is equal to 0.001 liter. Okay. So 200 microliter will be equal to 0.2 liter. So here we can write 0.2. When we calculate all of these values, we will get the answer to be 7.5 into 10 to the power 8. Okay, so here 7.5 into 10 to the power 8 is the correct option. So you have to remember this formula, that CFU is equal to number of colonies into total dilution factor divided by volume of culture plated in ml. ml is important here. Next is, in continuous fermented reaction, the phenomena of washout is obtained when? Okay, so let me tell you what is washout first. So washout solution describes the disappearance of population of any whatever we are growing. Suppose we are growing the yeast, so disappearance of the population of yeast cells in the fermentation. So whenever they disappear, whatever we are growing, if it is disappearing in the medium, then we call it as a washout. Okay, so washout is maximum, or washout happens when we increase the dilution rate. Okay, we keep on adding, suppose water we are adding. So whenever we keep diluting the culture, then we observe that the phenomena of washout. So the correct option is dilution rate is more than maximum specific growth rate. That means, suppose this is a glass of water. Okay, this is a glass of water. Now um, I am adding only little of the glucon D, suppose. Okay, and then keep on adding the water. Now what is happening? Now there is a less glucon D, and I keep on adding the water, so the taste of glucon D is going to be negligible. Okay, in the same way, the growth is very less when compared to the dilution. That means the washout happens then. Which one of the following compound has the highest phosphoryl transfer potential? Okay, so here we see this is phosphoenol pyruvate. PEP means phosphoenol pyruvate. So here it is having a phosphate group. It is having the highest capacity to transfer this phosphate group from this second carbon to ADP, and it turns into ATP then. Okay, and it is converted into the pyruvate. So here phosphoenol pyruvate is the correct option, which is having the highest phosphoryl group transfer. In the context of process control, stable system is defined as, okay, it is defined as one in which the output response is bounded for all the bounded inputs. This is the correct option. Next question is, the process of weakening the pathogen so that it cannot cause a disease is technically called as. Okay, see, opsonization means what? It is, this is a bacteria, and on this is an antibody. Now this antibody is a signal for a macrophage to come and eat this bacteria. Okay, this eats this bacteria. That means it is making the bacteria more tasty for the macrophage to eat it away. So here they are asking about weakening the pathogen. Okay, so opsonization is not the correct option because it is not the process of weakening the pathogen. Vaccination, no. Vaccination is not the weakening of weakening of pathogen. Next is attenuation. Yes, attenuation is the correct option, in which what we do, we slightly heat the virus or the bacteria. Okay, we slightly heat it so that it becomes very weak, that it cannot cause any disease in the body. Now when it cannot cause any disease in the body, we can put it in the body as a vaccine, and now our body can recognize it and produce antibodies against it. So this process of weakening the pathogen so that it can be used as a vaccine, those vaccines are called as attenuated vaccine. Okay, so the process is called attenuation. Next question is, polypeptide chains of the silk protein fibrin are predominantly in the beta sheet conformation, has a higher number of following amino acids. So in the polypeptide chain of silk protein fibrin, the majority of the amino acids present in the beta sheets. We have to remember beta sheet, because in the alpha sheets there is no presence of glycine. Okay, here they are asking beta sheets. In the beta sheets, glycine and alanine are predominantly present. Okay. Xeroderma pigmentosum is associated with the defects in. So this is a conceptual question again. Here, whenever there is the failure in the nucleotide excision repair to the damage of DNA, it causes a disease called as Xeroderma pigmentosum, which causes the pigmentation of the skin. Like what happens is, the pigmentation is lost in the skin, and there are white patches formed on the body. And that person, if he goes into the sun, uh, he becomes reddish in color. Okay. Next question. Which of the following is an antigen presenting cell? So antigen presenting cells are all of the three: B cell, macrophage and dendritic cell. So all of the given options are correct. Shrinkage of the protoplast from cell wall due to the exosmosis caused by hypertonic solution is called as. Okay. So the solution. Excuse me. Okay. This is a solution and this is a hypertonic solution. Hypertonic means, it means there is a less concentration of salt or anything we are taking. So suppose we put a cell here, suppose the RBC we are taking. So when there is hypertonic solution we are taking, what happens is there will be the movement of solute from the concentration of low concentration to the high concentration. That means what is going to happen? It is going to get filled into the cell, and this cell is going to now bulge. Okay, so what when this will blast, and what will happen? That process is known as plasmolysis. Okay, so second option is the correct one. Okay, it's asking, okay, I'm sorry, hyper solution, okay, hypertonic solution, na? Sorry, I'm so sorry. It is, okay, so solute always moves from the low concentration to the high concentration. Okay, so here we have taken the hypertonic solution. That means there is a less solute concentration here. Okay, so what happens? And here there is a more solute concentration. The solute is going to move from the solvent to the here, from the solution into the cell. Okay, so what is going to happen? It is going to bulge and lyse away. Yes, coming to the next question. Alpha helices in the protein usually do not contain certain amino acids which are called alpha breakers. Identify the correct pair of amino acids that is generally not found in alpha helices. So here the answer is glycine and proline. Glycine, because here glycine is having so many angles of rotation. Glycine is having so many angles of rotation. When it is having so many angles of rotation, it is not possible for the alpha helix to be stable because of it. And proline, because it is having a rigid structure. Okay, alpha helix need to be stable. So whenever there is glycine and proline, they do not favor the formation of alpha helices. So these are not usually preferred. So which one of the following statements is true for the reversible competitive inhibition of enzyme catalyzed reaction? So the correct option is the value of Michaelis-Menten constant increases. Okay. The bond length of peptide bond is, this is again a conceptual question, it is 1.32 Armstrong. Okay, here it is 1.32 Armstrong between the carbon and the nitrogen. The bond is called as peptide bond and the bond length is 1.32 Armstrong. Next is, which of the following statements does not hold good for the microbial production of penicillin by Penicillium notatum? So here penicillin is a secondary metabolite. Yes, penicillin is a secondary metabolite, but it is not asking not hold good for the microbial production of penicillin by penicillium notatum. So it is not at all related with it. So penicillin, okay, this is wrong. The process follows growth associated product formation kinetics. So yes, this is a correct option because this is actually a problem while producing the antibiotic. Okay, so the process is carried out by fed batch mode. Yes, this is correct. So this is the wrong option. Penicillin is recovered from the fermented bowel by solvent extract. This is also correct. So this is the wrong option. So because they are asking not hold good. Next is a fermentation broth with viscosity of 10 to the power minus 2 Pascal and density of 1000 kg meter cube is agitated in 2.7 meter cube baffle tank using Rushton turbine with a diameter of 0.5 meter and stirred speed of 1 second. The 1 per second. The impeller Reynolds number for the vessel lead. So the formula for Reynolds number is equal to, it is given by a little big formula that we have to remember to solve this kind of questions. So Reynolds number is equals to rho UL divided by mu, where rho is equal to density of fluid, U is equal to flow speed, L is equals to characteristic linear dimension, whereas mu is equal to dynamic viscosity of the fluid. Okay, so when we substitute all of these values, we get the answer as 2.5 into 10 to the power 4. So you can note down this formula and you can easily solve it. In the presence of competitive inhibitor Km of an enzyme, so what happens in the case of competitive inhibition? Suppose there is an enzyme here, okay? So one enzyme is having an active site. It is having an active site. So here comes and binds the substrate. Okay. So whenever there are more substrates. Suppose I have enzyme here. Okay, and there are 2, 3, 4, like these so many substrate. So all these substrates are going to compete with each other first. They are going to compete and then any one of them is going to come and sit here. So because of this, what happens is there is an increase in Km value. Km value is half of the value of the substrates occupied. So like there will be a time because they are already competing among themselves and then they are binding to the enzyme. Okay, so the Km value increases in this competitive inhibition. The main role of carbohydrate in the cell membrane is, so in the cell membrane the carbohydrate are always present on the outer side. We will not observe no carbohydrate inside. Why so? Because carbohydrate are helpful in the cell to cell recognition, signal relaying. All these happens outside of the cell. So that is the reason why carbohydrates are always present outside the cell and it is useful for the molecular recognition. Okay. Which of the following is correct for double-stranded DNA? Purine is equals to pyrimidine. Yes, this is correct. AC plus GT. Yes, this is A pairs with T and G pairs with C. A double bonds with the hydrogen bond with the T, and G is having triple bond with C. So here AC is equals to G, A plus C is equals to G plus T. This is also correct. And C plus T is equals to A plus G. That means purine is equals to pyrimidine. This is also the correct option. So all of the given options are correct. Most of the mitochondrial DNA is inherited by. So mitochondrial DNA we only inherit from our mother. Okay, so here stem cells is wrong, sperm is wrong, neither sperm nor egg is wrong. So X cell is the correct option because X cell is the mother cell. We receive the mitochondria from the mother cell, that is X cell. Next is to promote the attachment and spreading of anchorage dependent animal cells. The surface of the culture vessel needs to be coated with. Okay, so here the correct option is pronase. So a pronase is also known as a dispersive agent. It's also known as a dispersive agent of tissue culture. That means whenever we put the pronase onto the medium, this will encourage the spreading of the cells onto the medium. That is why it is known as dispersive agent. Pronase is the correct option. The process in which microbes are used to reduce the toxic industrial waste or effluents is termed as. So the answer, this is conceptual question again. This is known as bioremediation, in which the microbes are actually used to remove the pollutants from any waste, like soil, water, environment. So that process of removing the pollutants is known as bioremediation by using the microbes. Which organelle is not present in an isolated cell protoplast? Okay, cell wall is a correct option because see, this is a plant cell. Okay, this is a plant cell. It is having a cell wall. So whenever we remove the cell wall using any enzymes, okay, like cellulase, okay, we have only the plasma membrane cell. Now we don't have the cell wall. So a plant cell without a cell wall is called as a protoplast. So protoplast is actually used for the culturing of plant cells. Which one of the following interactions are involved in the stabilizing the structure of protein? Hydrophobic interactions, yes. Ionic bonds, yes. Hydrogen bonds, yes. So all of the given options are correct. Next is hypophosphatemia is inherited as sex-linked dominant disorder. A normal male marriages a heterozygous female affected with hypophosphatemia. That male is normal, that means XY, it is normal, is crossed with a female who is affected. That means it is having the gene on both of its X chromosomes. So now, what proportion of the offsprings will manifest the disease? So let us do the cross. X, X, and then we have, sorry, male is normal, so we don't have it here, and Y. So let's cross it. It is going to be X, X, this is will be affected, X, X, this is going to be affected, X, Y, this is affected, X, Y. So heterozygous female, okay. What proportion of the offspring will manifest the disease? So you see, everyone here are getting the diseased allele, that means X-linked allele, because female is having it on the both alleles. So 50% of the sons and the 50% of the daughters are going to show the disease. This is a 50% and this is another 50%. Sorry, this is the female, this is a male. Okay. Next question is, base sequence variation between individuals that result from point mutation are called restriction fragment length polymorphism. No. Single nucleotide polymorphisms. Yes. See, every individual differs from each other. Suppose I have A, T, G, C. Okay. If another person is going to have A, G, G, C. So there is only difference in the one nucleotide, and this difference is called as single nucleotide polymorphism. Okay. So that's all in this video, friends. In the next video, we will be discussing the question from 61 to 100. Okay, thank you.